Grand, I think we'll make a start. Good evening, everyone, and welcome to this Farm Advisory Service webinar as part of the Sustainable Sheep System series. My name is Daniel Stout, and tonight we'll be discussing Johne's disease in sheep. Tonight's webinar, then, we're going to run for about an hour until about 8:30. We're joined by Dr. Craig Watkins of the Moredun Research Institute. Craig is a leading expert on Johne's disease. Just before I go through his biography, just to clarify, Johne's disease, also known as paratuberculosis, is caused by the Microbacterium avium subspecies paratuberculosis, also called MAP. So we're going to hear the phrase MAP quite a few times tonight. So Craig has a degree in microbiology from Aberdeen University and has worked at the James Hutton Institute in Dundee before attending a DPhil at the University of Oxford. After a postdoctoral position on gene therapy at Leeds University, he joined the Royal Dick School of Veterinary Studies at the University of Edinburgh for the next 5 years, working on orth and also starting out his career of the focus of Johne's, looking at host responses to sheep, specifically in sheep. Craig then joined the Moredun Research Institute as a postdoctoral scientist in 2003, and over the past 20 years, his interests have been applied molecular approaches to the understanding of host immune responses to disease and bacterial genomics with respect to MAP. Recently, his research has extended to studying the microbiota of ruminant intestine infected with MAP and/or co-infections with parasites. Craig also supervises a number of postgraduate students and is closely involved in preparing and presenting science to the public through STEM and also the Moredun's communications team. His current research, which I think there's a lot of interest here, is looking at the functional studies of MAP genes for vaccine development, looking at immune responses to MAP infection, DNA sequencing of the intestinal microbiomes in sheep with different health conditions and assessing their associated environmental microbiota, and MAP transmission and contamination in milk and the environment. And finally, looking at on-farm MAP control measures to reduce infection and improve welfare and productivity in the farming industry. And I think we're excited to kind of hear a bit more about that and the latest research tonight with Craig. Just before we start, I'd like to kind of flag up our upcoming webinars as part of the Sustainable Sheep System series. So we're back next week with Professor Laura Graham of Birmingham University to look at lameness and foot rot. We're then going to be back on the 5th of December with Professor Richard Wall looking at blowfly strike. Then following Christmas, we'll be back in January having a focus on ewe lamb selection. And then finally, to wrap the whole series up and kind of tie in that genetics and the health aspects, we're very excited to have Dr. Mark Ferguson from NextGen Agri in New Zealand over to talk to us via webinar about the research that he's been involved in, looking at foot rot and blowfly strike, particularly merinos, and actually the development of EBVs to breed for reduced susceptibility or improved resistance to these diseases. So we're excited to have him on the 20th of February 2024. And just to say, I will send a follow-up link to everyone tonight with the sign-up links to all these different webinars, and I'll keep you updated ongoing as we have links for the later two. I'm sure many of you are familiar with Zoom now, but if you've got any questions, you'll see the box at the bottom. We would ask that if you've got any questions for Craig, we'll have plenty of time for questions at the end. I would ask you to put it in the Q&A tab, but if you've got a more private question, a question about maybe the tech not working for you, please put it in the chat box and we'll try and help you with that. And bear in mind that we are recording this webinar, but that will mean that it's available on YouTube later on. This slide's always quite slow. We just ask you to please fill in the feedback form when it comes after the webinar. It really does, it does make a difference. It helps us improve. We get your feedback. It helps us identify opportunities and the gaps that you want to learn about. And it also helps us feedback to Scottish Government so we can continue getting funding to deliver these webinars and other knowledge transfer outputs. So I will now pass you over to Craig. Grand. So I'll let you take it away, Craig.
Yeah, thanks, Daniel. Thanks so much for the nice introduction, and welcome everybody to this evening. I really hope that you can learn something from it, but also hopefully we can have a really nice open discussion afterwards. And as Daniel was saying, it'd be great if you guys could just add in things in the Q&A session as we go along, yeah, hopefully we can have a really good discussion. So I'm just going to share my slides now, and we'll get stuck in. Okay, so As Daniel said, we're going to be focusing in on Johne's disease in sheep tonight. So, but I dare say there's a number of you out there that not only farm sheep, but other livestock as well, probably cattle. So I'll be touching on a few other species of livestock as we go through the talk. So just to start with, I just want to introduce you to where I work at the moment, and I have been working since 2003. This is the Moredun Research Institute now, just south of Edinburgh. It's it's around about 6 miles south of Edinburgh, and it's in a bigger campus, science campus, which includes the vet school, the new vet school, SRUC, the Roslin Research Institute. So there's a whole pool of us, all in about a 10-minute walk of one another, and it's a nice site to work in. It's the Moredun Research Institute is under the umbrella of the Moredun Foundation, and then there's a couple of departments within the Moredun Institute: the Vaccines and Diagnostics department and the Disease Control department. We also have a spin-out company called the Moredun Scientific, and the Pentland Science Park runs the whole site. And then we have a number of little spin-out biotech companies that are in these smaller buildings around the outside. But we're in this larger building here. And we're just over 100 years old. And 100 years or so ago, we were in this little, well, I guess, lorry here, really. That's how we all started off, in a lorry, not myself personally, but 100 years ago, this is how Moredun started, in a van, going around various sheep shows, trying to help farmers to understand some of the diseases and infections that their sheep were getting. There's a lot of labs, but there's also a lot of animal containment in the Moredun Research Institute, and lots of containment facilities as well. And we have about 120 staff, and and we work on lots of different diseases of both, well, not just sheep, but cattle and pigs as well. This is our specific team, so I'll be going through a lot of research that we've been doing, and these are the people that have been carrying it all out. So you'll see me in different guises here. So this is us just last week on a farm. So this is Rachel Richardson, our research assistant, and Charlotte Winspear here is our PhD student. So you'll see a lot of the work that they've been doing, along with this group over here, which we work with so we work in collaboration with lots of different people on Johne's, and in this particular case, working on the farms, we work with James Hutton, the Aberdeen group, with the lead being Yulin Pagaling here in the red. We also work with other people within the Moredun Research Institute, and that's particularly the parasitology group, and that's led up by Dr. David Bartley. We also work with the University of Edinburgh and Dr. Andrew Free. There's one other person in our mycobacteria group, Dr. Valerie Hughes, and she works primarily on the diagnostic test improvements as well. Of course, we always work very closely with our vet practices and our farmers as well, and it's to them that we owe a lot of gratitude, and thank you for all your patience if we've ever been on any of your farms to work on a Johne's project. So let's get into what Johne's disease really is. As Daniel mentioned, it's caused by this bacteria, Mycobacterium avium subspecies paratuberculosis, which is rather a long set of words. So we're just going to call it MAP for the rest of the evening. It affects ruminants predominantly. So in our case, what we work on are cattle and sheep, but also deer as well. Young animals are the most susceptible, so when they're newborns, and transmission can be and is usually through the fecal oral route or through contaminated milk, but it can also be through the in utero and in semen. The interesting thing about the infection is that, of course, it stays subclinical for a long time. There's often no sign that the animal is diseased, even though they can be infected. And it may be that they will never get any kind of clinical disease at all, but will be infected and may also be able to transmit the disease, even though, you know, that they seem to be in good health. What are the clinical signs? Well, hopefully you won't see too many animals looking like these. This is kind of the extreme end of the condition, unfortunately. Loss of condition, emaciation, and diarrhea. So diarrhea in sheep particularly is not necessarily indicative of the clinical disease, but they can have diarrhea as well. The cattle often will have diarrhea, and in some cases they will be classed as super shedders, in that they actually shed a very high amount of MAP in their feces, and this can be a really big problem for transmitting Johne's disease between cattle, and also between cattle and sheep or deer. So just to be aware that sometimes animals will shed a large amount of the bacteria, but at other times they may not shed any detectable level of MAP. So tricky thing sometimes to diagnose, which we'll get onto in a minute. The pathology. So once we do postmortems, and the vets will do postmortems as well. And this is a picture of the small intestine. And you can see this really corrugated kind of look to the inside of the small intestine. And this is a healthy one here, and this is one that's been infected with MAP. Over here on the right-hand side, you'll notice that this is very orange, and this is quite interesting because very often what we find in sheep, the sheep strain of MAP, is that it's got a pigmentation of being either yellowy or orange in the gut, and so that's really easy to work out what type of MAP isolate it is, whether it's a sheep one or maybe a cattle one. It's not 100% proof, because sometimes we get sheep strains that are not pigmented in this strong orangey yellow color, but a lot of the time we do. What is the organism? So the organism has been completely sequenced, and so it's just under 5 million base pairs. This is what it kind of looks like. It's kind of a nice rod shape. This is a scanning electron micrograph of the Mycobacterium, or MAP, and you can see it's kind of like a tube or a bean, I suppose. There. This is what it looks like inside a cell. So all these purple stained things are the MAP within the guts lining. So this is the edge of the gut along here, and then you can see that it has infiltrated into the gut lining and into cells called macrophages, and these are the cells here. So you can sort of see the outline in red, and then the blue is the nuclei. The little green bars here are the bacteria. They're mycobacterium dependent, which means that we have to add that to any cultures we grow, and we call them acid fast because we use an acid fast stain to make them go this kind of pinky purple color. And as I said, they're something that grows inside macrophages, one of the immune cells in ruminants. So how resilient are they? They're obviously a bit more complicated than just normal bacteria. Mycobacteria have this really complicated outer coat, which I've just kind of shown as a schematic here on the right. But it does mean that they are pretty good at surviving in the environment. So there are studies now that have shown that they will be perfectly viable after 9 months in water, 11 months in slurry, and up to 47 months in soil, depending on, of course, the soil type and the conditions. They're also quite resistant to pasteurization, chlorination, freezing, and a lot of antibiotics. So prevalence and cost. So it's a tricky one, this prevalence, because we haven't really done much in the way of prevalence studies in the UK. There's been more perhaps in Australia and New Zealand, where the sheep industries has been going for quite some time. It's quite large. But in the UK, we haven't really done very much. This came out in almost just over a year ago now in the Farmers Weekly from the University of Liverpool, from Dr. Piers Davies, and it it seems to suggest that actually that Johne's is ovine Johne's in sheep is present between 59 and 65% of farms surveyed in the trials that the academic and industrial trials that they've looked at, which is quite high. And so I would say that, and a lot of my peers and vets I've talked to have said that, really it is one of the big underreported diseases in sheep particularly. In cattle, slightly different story, but in sheep, quite underreported. But just to give you a bit of an idea, this is slightly older data, but it gives you a bit of an idea about the spread. So in the map on the left here, the blue dots represent the diagnosis in cattle in Scotland, and then you'll see the quite underreported cases of sheep as well, and they seem to overlay a lot of the areas where the cattle are as well, and that's something to be to consider, because as I said before, the strains that infected cattle also infect sheep and vice versa. So we're going to move on to one of the big areas, the big issues in Johne's disease, and that is the testing. And I think we all, and I can certainly hold my hand up and say it can be very frustrating and sometimes a little bit confusing as to what test means, what the tests mean, what the test results mean, or what the tests are for. And there are a number of tests, but as you can see, there will be limitations to each of them. One of the things about it, why it's so difficult to diagnose, is because it's got this really long incubation period. So you can have an infected sheep for quite a long time, and it will show disease for maybe 3-4 years, and then but will remain silent in this sort of subclinical infection stage for quite some time, but will still shed the bacteria through its feces and milk, even if it's not showing any physical signs of the disease. There is a immune response change which happens though between this sort of subclinical phase and the phase where they become clinical, and that's one of the areas that we've been working on a lot in Moredun, and also with our collaborators across the world, as to how that immune response changes and why it changes to push the disease into a much more clinical state. This is one of two graphs I'm going to show you tonight. So it just gives a little bit of a nice summary about what's going on in terms of the immune response. So as you can see with the fecal shedding here, this is the black line, over time, you can see that the fecal shedding does slowly increase. It's kind of on and off up to a point where you go past this line, which is where you're starting to go into more of a clinical phase. So this dotted line here. So once you get into a clinical phase, then fecal shedding can often be fairly regular, but still not always consistent. But obviously in the sort of subclinical phase, it can be very, very sporadic. The antibody response, which is something that we use and have used for a diagnostic test, the blood test for Johne's, you can see that the antibody test, the antibody levels actually are only high once the sheep becomes clinical and starts showing signs of disease. One of the interesting aspects of the disease is this interferon gamma response. This is a cytokine, an immune response, which happens really early in infection. So this is the red line here, so very early. And we've been trying to work in our group and in other groups to try and harness this to see if we can use it for an early diagnostic for Johne's disease. So there is no single test at the moment which will reliably detect all stages of disease and the subclinical carriers who can still transmit the disease. And of course, a negative test result doesn't always mean there's no infection. So that's the complication of the testing at the moment. So which test? So we need to know what the purpose of the testing is. Is it for individual testing, herd testing, or flock testing? The animal species being tested, the age of the animals. So as I was mentioning before, the serum antibody test may not be very sensitive when you're maybe testing really young animals, because that antibody response has not really kicked in until they're a lot older. The Yoni's history of the flock, and then of course the accuracy. So are you going to use it for an individual animal or a flock? And of course there's the economics to consider, you know, the cost of all these different tests, and whether of course you know, how costly is a false positive or a false negative result as well. These are some of the tests over here. So this is an ELISA assay, which we can use. There's the qPCR, which is generally used for looking for the DNA of the genomic DNA of MAP in feces and in milk and semen as well. And this is our culture. So this is how we can grow MAP in the lab. And of course, if you can get the bacteria to grow from a sample, then you're pretty much convinced that that particular animal is infected. That's kind of what we've always called our gold standard, but it's not particularly sensitive. It can be very specific, but it's not very sensitive. So a negative, again, doesn't always mean it's a negative result. How can we improve diagnosis? So a careful choice of the current diagnostic tests. So I've mentioned the qPCR and the serum antibody test. Those are two of the main ones at the moment. And using complementary tests, and consider maybe using the test not just once, but several times throughout the year, perhaps, if you think that you've got a serious case of Yoni's outbreak in your farm. Because as I was saying before, for the fecal tests, yeah, sometimes MAP will be being transmitted through the feces, sometimes it won't. So you can miss it on a single test. So with all the difficulties of this diagnostic test, why control it? And so I just thought we'd go through this next section of the talk and just say, well, why control it? And what are the risk factors? What can be done about it? Okay, and what are the options for control? So the options for control would be for those who don't actually have Yoni's disease at the moment on their farms and want to keep it off, obviously, but also for those who have had positives on their farm and want to know how to control it. So why control Yoni's disease? Well, there's lots of different issues. There's also obviously the welfare issue to the animals, the productivity as well, and the financial losses to the farmer. But there's also possibly a public health issue with the link with Crohn's disease. So we know that pasteurization is quite resistant to pasteurization. We know that it can turn up in raw milk, obviously, and in cheeses. But also there is this link to Crohn's disease. And although there's no direct evidence as yet to say that paratuberculosis or MAP causes Crohn's, there's lots of associated studies, so association studies, which suggests that you know, that there may be an effect there. We've yet to the researchers within that area or that field of looking at whether MAP causes Yoni's have yet to conclusively prove that's the case. But there's anecdotal evidence that suggests that there is a possible association. So that's something to bear in mind. And I know that that dairies and some countries are very aware of this issue as a sort of public health issue. And also, of course, if we don't do anything about the disease, like some of the other infectious diseases, they only get worse. So I just want to introduce you to this idea of the iceberg diseases. So there's a number of, there's about 5 different iceberg diseases, and I think you've had a talk previously, perhaps, about other iceberg diseases. OPA, for instance, is an iceberg disease. And basically, if you haven't come across this idea before, is it just that if you see an animal that's got some really bad clinical disease, is wasting away, it's come up positive for Johne's disease. If you get one or two of those, chances are there's maybe between 15 and 25 of your animals that are infected, but are in this sort of silent phase when you can't really see any symptoms. But as you go up this pyramid, obviously you begin to find that some are carriers and they can then begin to spread the disease. So here you go, low to medium shedders at this stage or phase 2. So this is what we call an iceberg disease, and it is quite difficult to manage. It can be quite difficult to manage. However, as I was saying before, if you do find that you have got Johne's on your farm, and you want to try and do something about it, doing nothing is kind of in this red line. So this is my second of my 2 graphs for tonight. So bear with me. So the x-axis, this is the years over years, and then percentage of flocks showing clinical signs, or that, you know, when they're definitely showing disease, up this axis here. So if you look at the red line, this is what happens when there's no control program put into place on a farm with Johne's, and then and it goes right past this sort of 10% line here, which is 10% of the flock, for example, would be infected and showing clinical signs. However, if you put in a test and cull procedure, you can see that it does have a bit of an effect. But as you can see, it kind of tails down to maybe about a 5% sort of residual infection rate in that flock. And that can carry on for many, many years, as you can see, between here, which we're saying around about 11 years, all the way on to 30 years. There's some management changes you can put into place. So to try and isolate animals, to try and keep pens clean and things like that during lambing, that sort of thing. Again, it will have an effect, but again, it's not going to happen very quickly, and it'll take up to, you know, over a decade before you can actually really see a big difference going on. But by combining the management changes, a test and cull, and perhaps vaccination program, you can see this yellow line, it seems to reduce over maybe 5 years. 5 years does seem like a long time, I agree, and we wish we could do, and I'm sure the vets wish they could do better than that to try and curb this disease at a faster rate, but at the moment, with the tools we have, that's probably the best option. So what are the risk factors? So in terms of biosecurity, it's all about trying to keep it off your farm if you haven't got it on your farm, and that's to do with the new stocking. So that's can include semen and embryos. Management practices, so it's about managing your colostrum and your waste milk, manure, and water. And of course, the environmental side of it is, again, water sources, other livestock, and wildlife reservoirs. So what can be done to reduce the risk? So this is kind of you know, the sort of practical applications that we can put into place. And developing a health and welfare herd or flock plan is really, really good in conjunction with your vet. A health scheme, getting involved in a health scheme, and I'm sure a lot of you are aware of these kind of health schemes. There's certainly the premium sheep and goat health scheme available, and some of you will be aware of the cattle ones as well. And then just, yeah, implement some really good management practices to limit the spread of disease. The other things in biosecurity is trying to reduce the amount of sheep and animals you bring in on the farm. So maintaining a closed flock is a really good idea to stop a disease entering into that flock from other sources. But of course, when you do need to bring in animals, then to try and source them from a source that has been confirmed negative for maybe the last 3 to 5 years through an accredited flock health scheme. If larger numbers, of course, are required as replacements, then and you are looking to expand your flock, then the suggestion is to try and buy them from a single flock and test that whole flock in quarantine before you let them out into the rest of the farm. And then consider screening your cull sheep, your poor yielders, every year, just to see what kind of diseases they might be struggling with or are being affected by. And it may not be Johne's, but it may be something else that you can actually do something about as well. So it's well worth just screening occasionally these cull sheep, just to get them tested to see what they might have, and it may well be that they could have Johne's as well. So you might be able to catch the disease at an early stage at that point. So culling clinical cases is quite important, as I was showing on that second graph, and the offspring of those infected animals, really important not to put them back into your breeding flock, and then keeping lambing pens clean and using disinfectant, and that goes for most infectious diseases. Rearing lambs, of course, in clean environment free from fecal contamination is really important, as again, not just for Johne's, but for other infectious diseases. And avoid pool colostrum or discarded milk. So if you've got Johne's on farm and you have one particular sheep that is producing a lot of Johne's in their milk, and then you pool that, then that can spread, of course, to other lambs. Providing clean drinking water is really good. So we're moving on to the kind of environmental side of the risks. So fencing off stagnated water. So this is not only great to try and reduce Johne's, but also other diseases. I'm thinking of liver fluke, for instance, and reducing where the snails might reside in puddles or stagnant water and pools, and keep the old water troughs cleaned out as well. With slurry management, it's quite important to make sure that and try and get the manure in some way treated, so you can compost it for quite some time, and the heat in there will actually reduce the amount of paratuberculosis or MAP in that manure if it is contaminated with MAP. And if possible, and I know this can be really, really difficult and a real challenge, but to avoid co-grazing or sequential grazing with infected stock. So that would be sheep that you may not be sure whether they are infected, or they may be infected, and not try not to mix them with your other flocks, but also try not to co-graze or sequentially graze cattle, for instance, that may be infected with Johne's as well. And I talked about wildlife reservoirs. What I meant by that was wildlife that might be able to carry MAP, the causative agent of Johne's, and we now know for certain that there's a number of these wildlife reservoirs, and in particular the rabbit. So quite tricky sometimes to manage this disease when the rabbit population is going well on a farm, and it's known that they actually have Johne's in their feces. So what are the options for control? So we're kind of aware of the fact that there is this vaccine available, which is quite affordable now. It's the price of it's gone down, and you'd have to ask your vet the cost of that. But it's a one shot, 1 mil, one shot for the life of the animal. And it does prevent the clinical side of the disease, and does show to reduce the amount of the bacteria, the MAP, being shed in either feces or milk. But it has a few drawbacks. So it doesn't prevent infection. It just reduces the possibility of infection in the sheep that are vaccinated. It stops you from being able to use the antibody test, the blood test, because they all come up positive because they've been vaccinated with this vaccine. So we can't differentiate between those that are vaccinated and those that are infected using this particular blood test once you've vaccinated. It also interferes with the bovine TB test. That's a bit difficult, and there's no differentiation between the vaccinated and, as I said, between the vaccinated and the naturally infected animals. The other thing is that, unfortunately, because it does interfere with the bovine TB test, it's not recommended for cattle. So I'm just going to move on now to describe a little bit what Moredun's up to in terms of MAP at the moment. So as I was mentioning before, Dr. Valerie Hughes works primarily on the diagnostic side and looking to understand these kind of early immune responses and trying to identify some new biomarkers for the disease and to try and improve these diagnostic tests so that maybe we could get an earlier diagnosis and maybe to try and understand why it is that some animals might be infected but never transmit the disease, and some might be infected but never get clinical signs or symptoms of the disease. There's also the vaccine side. So I work a bit on the vaccine side along with Rachel Richardson. And so we've been working on a new system, this new organoid model of infection, which I'll just briefly touch on in a minute. And also at the moment, we're working with Charlotte Winspear, and the team are all working on this kind of environmental side of things as well, looking at the microbiome within the fecal samples of the sheep, but also in soils and water on the same farms. And we're also looking at these co-infection studies. So I'll just move through to just show you what I mean by all these different projects. So this is the organoid. So we can actually grow what we call like mini guts in the lab now. So these kind of are much better at mimicking the infection in the small intestine, and we can grow them quite effectively under very special conditions. And this is just a schematic kind of show you how that works, and then you can infect them. And this is a kind of infection here. This is these organoids here. The little blue dots are the nuclei of all the different cells. And then as you see in this higher magnification, that's a cell there with the nuclei in the middle. And then when you stain them for the map, you can see all these yellow things. Now, yellow dots and yellow little bars. Some of them are in little single bacteria. Some of them are in clumps because the bacteria actually does like to clump together. But that's showing you a little bit about how we're getting on with the organoid work. And we're trying to use this system to understand a bit about how they infect the surface layer of that small intestine, but also to try and find new vaccine candidates. The co-infection studies have been going on for quite some time, so we know that livestock diseases don't occur in isolation. In the past, we've often worked with them in isolation as researchers, but actually we're very much aware that diseases don't occur in isolation in sheep, particularly, or livestock generally, and that they can affect tests and treatments and vaccine efficacy. And these are really poorly understood. And we've been working on with the parasitologists at Moredun under David Bartley to understand what the association is with roundworms, particularly in the small intestine. And so we've been using a cell culture model to have a look at this and how they affect one another, the MAP infections versus the roundworm infections, and in combination. We've been looking at it through microbiome studies and we'll be looking at that as a sort of vaccine efficacy trial, which we've done, but we haven't actually done the microbiome studies on that yet. But we're trying to get the analysis finished. And the treatment for roundworms as well, whether the efficacy of those drenches are affected when the sheep are also infected with MAP. The next thing we're working on, which is quite exciting and quite new to us, is looking at sock sampling. Now, this is what we think might be a very cheap and effective way of finding out what bacteria that might cause disease are on a farm. That would include Johne's disease. So this is not completely novel because I think there was a study back in just at the end of 1999. I think there was a study with salmonella in chickens in chicken barns, And we're trying to use this study to try and see whether we can mimic what we see on these areas where there's a high trafficking of sheep here and around food bins and things like that. And we're trying to pick it up using these sponges, and then we extract all the DNA off these sponges, and then we can identify from the DNA what type of organism might be there in the farm. And we're going to try and compare that with what we find in the sheep flock. So we are also testing a good percentage of the sheep flock at the same time as we're doing this, and to see whether we can pick up similar microbiome profiles, what we call microbiome profiles, from these different samples. So that's in progress at the moment. So this is Ali Morris from Moredun and Amy Wallace as well, a previous research assistant who used to work at Moredun. So as you can see, there's quite a bit we can do to manage the disease, but there's a lot of unanswered questions, and these are some of the ones that we are at Moredun and other people around the world working on UNIs are aware of. You know, things like there's lots of different types of MAP. There's the sheep strains, obviously, and the ones that go yellow, as I mentioned earlier. There's the cattle strains. There's lots of different sorts. And are they all equally infectious? Do they all cause disease? We don't really know the answer to that yet. Is there a minimal infectious dose? So how many bacteria are needed? How many MAP are required for an actual infection to take place? And how many would be needed for it to kick in a full-blown disease? And are all ruminants equally susceptible? We're not entirely sure about that either. There's been quite a few studies about trying to figure out whether specific breeds are more susceptible, but the jury's still out on that. What factors control infectious progression and the onset of clinical disease? Again, that's very much an immune question at the moment that we're trying to understand. How do wildlife reservoirs impact on our control measures? Well, we know that they can do, but to what extent, it's hard to know. And then husbandry practices. So how does management practice affect transmission? And then, of course, we're back to the co-infection studies that I've just mentioned, and whether that have an effect on the outcome of infection. And we really do want to know and try and design the perfect diagnostic test, which would allow us to understand, when we get the test result, what's going to happen to that animal. Are they infected? Are they not infected? Are they going to progress to transmit the disease? Are they going to go and progress to actually getting clinical disease? And if we could get a test that could answer those kind of questions, then I think we'd all be in a much happier place. So thanks very much for that quick tour through the Johne's disease, what it is and how we can manage it more effectively, and also sorts of things that we've been working on at Moredun. And this is just to let you know that we work very closely with the University of Edinburgh and other members of the Moredun family, but also the James Hutton Institute. And of course, we're funded by the Scottish Government, as well as some of the projects I've mentioned are also through the East Bio PhD studentship program. And I'm hoping that you've had a chance to think about some of the questions that you wanted to ask as we went through that. And I look forward to discussing this in the next few minutes. Okay, thanks very much. I'll stop sharing my screen now, and I'll hand back to Daniel.
That was great. Thank you very much, Craig. It's I've certainly learned a lot there, but it sounds like something that the deeper you, or the more you know, almost the less you know. There seems to be so many unanswered questions on the topic. And as such, we've got no lack of questions coming through here as well.
Ah, good. Okay.
I suppose the first question was about, you know, you showed the graph about sort of distribution, but do you think that there is less Johne's in sheep than cattle, or is it just simply that we don't test sheep as much?
Yeah, that's a good question. Thanks for that. I think the thing is that we don't test as much in sheep. I think partly because the value of an animal, perhaps, as well. But we've always been we've always been more aware anyway that the beef sector tests more, and certainly the dairy industry too. So I think it's just underreporting. Less testing is my feeling anyway.
Could also the systems that they're running in terms of being more housed also
Yeah.
have an impact there? You know, a lot of sheep are more extensive. More
Yeah,
indoors, I
absolutely.
suppose.
Yeah, absolutely, Daniel. Yeah, I agree. That can definitely have play a factor in whether things are tested or sheep are tested or not. Yeah.
Got another question here. If you've got yonies in your cattle, many people will know because they test. Will you almost definitely have it in your other species? Are they completely cross strains? Is there different strains in any fashion, or?
Yeah, that's a good question. I mean, I think we've certainly found evidence whereby the cattle strains will move into the sheep, and we have come across it working in reverse. Interestingly, deer are particularly susceptible to Johne's disease, and they can produce a lot of MAP in their feces as well. So people may be farming deer as well. Certainly, there's been a lot of studies of this in New Zealand, obviously, because that's where they do a lot of farm deer. They have a big farm deer industry. And yeah, so they get very severe clinical disease, and so it can be transmitted between the cattle, the deer, and vice versa, and then of course onto the sheep as well. So it's a very dynamic process. I think they do move. The different strains do move between the different species of livestock. I think if from a research point of view, the sheep strains are much slower growing for us in the lab, and they may well be a lot slower growing in the animal, but we're not sure about that.
Because what would be the typical ages that you would see it in cattle versus sheep in terms of clinical signs?
Yeah, so that's interesting. So in cattle, we find that there's a sort of a linear progression, whereby the cattle will be infected, and then they'll go through what we call a sort of paucibacillary stage, and then a multibacillary stage, which is when they become clinical, and will die from that. But it has to be remembered that perhaps only 10% of herds might, in cattle anyway, would actually come down with clinical disease. So the rest of them may well be hidden, which is why I mentioned the idea that it's one of these iceberg diseases. And but for sheep, it's slightly different, or can be slightly different in the fact that you can have sheep going down, getting infected, and then they kind of can go off in two different directions. So they could go just down this sort of paucibacillary route, which we call, or the multibacillary route. So they can still transmit the disease through their feces and through their milk in both phases, or both stages, and can die from both those stages. But they look quite different when you look at the sort of histopathology of the tissue and the immune system. So their immunity does seem to be different depending on which path they go down. And we've done a lot of genetics to try and understand whether that's a breed susceptibility issue, why some sheep go down one direction and other go down another direction. So they are slightly different in the dynamics of the disease between the sheep and the cattle.
Yeah, it's really interesting, isn't it? We have plenty of questions coming through now. Go with this one here. Can a ewe be transmitting MAP in colostrum and milk without having clinical signs?
I believe so, yes. So the main route is fecal transmission. So that's the main route. But yes, it is understood that they can still secrete and produce MAP in their milk, even in a subclinical stage, but at a lower level, I would guess.
Okay. I suppose tying in with a similar question, if a ewe has Johne's, will her lambs definitely get it?
Sorry, say that again?
If a ewe has Johne's herself, will her lambs almost definitely get it? would you expect our lambs to get it each year?
Yeah, so we suspect that that's when they are the most at risk, is when as lambs, their immune system hasn't really developed very well. They're going to be really open to all sorts of diseases, obviously, but we are pretty confident that's when they're going to get it, if they're going to get it. And it is often, it can be just contamination of teats or contaminated milk that they contract disease from, but also through the colostrum and milk as well. So I would say that early stage is critical, and for a Johne's positive ewe, it's suggested through a lot of control programs, not to keep those animals and those lambs either, and particularly not for breeding purposes, and their lambs. Yeah.
And I suppose to that end, two parts to this, I suppose. If I have clinical signs that I suspect there's probably Johne's in my flock, what's the first thing I should do? What would be your sort of go-to test to kind of test the water, I suppose, and have a look in?
Yeah, so that's a really good question. I get that question quite a lot from various vets and farmers. What is the first thing to try and have a stab at? And I think one of the tests that is certainly used in cattle are obviously the serum antibody test, the blood test. The blood test in sheep is not particularly sensitive. It can be kind of used as a sort of herd screen, so you could maybe find out whether any of these sheep have actually been exposed to the infection. Gives you a bit of a broad sweeping kind of idea about what might be out there. But if you think you've got Johne's, so you're seeing what you think are clinical disease, you've tried maybe it's a worm issue, so you maybe done a drench or something to try and control the egg load from parasite infections, and that doesn't seem to have had any effect. Then try the qPCR from the fecal samples. That's well worth doing. What you can do is you can have these pooled, and so you can say group together 4 or 5 samples from your sheep, and then put that in to be tested under qPCRs through a surveillance program. So there's a couple of them. We certainly have SRUC and BioBest that do that work up here, and they'll screen those for you, and then you can identify individual sheep that are actually producing the bacteria in their feces. Now, that's not a absolute test, because obviously we find that sometimes the bacteria is not coming through the feces at all at that particular test time, and then it need to be repeated. If these sheep are really losing condition, then it might well be worth just having them culled and having them PM'd. So a post-mortem is really effective way of finding out whether you've got Johne's. So by looking at even just the gross pathology, but also some of these the histopathology of the area in which they infect the gut, that small intestine, and then you can get a real idea from that as to whether Johne's is in your flock or not. So individual sheep, when they're losing that kind of condition, if you can manage it, look at these cull ewes and just see what's inside them. Yeah,
brilliant. And so the next part of that would be when we know we've got it in the flock. You mentioned a sort of test and cull program, so that's individual animals. What would that look like? What tests would you do? How often would you test and cull?
Yeah, so it's a tricky one, and that's something I think you really need to work through with your vet. And obviously you've got your own management plans in place, and every farmer will be running their business in different ways. So it's a difficult question to answer, actually, Daniel. But you know, if you have got yonis on your farm and it's getting to the stage where you're getting a higher mortality rate, so you know 5-10%, then yeah, the testing, you know, frequently, more than once a year, just to try and get your head around what individual sheep are actually causing the disease, and trying to get them out of your flock. Really try to get them out of your flock as quickly as possible, and also just be kind of aware what animals, other animals might be there as well, other livestock. So some people might find that, you know, they get 1 or 2 cattle that come down every year with yonis, and they can't figure out where it's coming from. But if they've got sheep, then it might be coming, you know, through their flocks and things like that. So it kind of varies depending on your kind of farm makeup, really. But I would say, yeah, try to call them out when you find that you've got a positive, certainly a fecal culture positive, Daniel. Yeah,
sure.
If you really want to try and clear them out. And of course, then there is the other side of it, is the biosecurity side, and the vaccine.
Perfect. And this really is one to have a good chat with your vet about, in terms of options are appropriate.
Oh yeah.
I'll just draw people's attention as well to the Preparing for Sustainable Farming grant scheme at the moment. Would pay £250, and one of the options there towards that £250 is to do a cull use screen. So well worth looking at. You can get 2 options in the year, and if you do it this year, you can also get the sort of personal development pot of funding as well, which is £250 on top of the £500 for testing that you can do. Couple more questions here. If you vaccinate, will the offspring test positive, or would that be a way to start afresh as such?
Yeah, so the offspring won't necessarily go positive because you've vaccinated their ewe, the mother. The ewe will be positive when you do a blood test, but the lambs won't be vaccinated. So the thing to do is and we've done this on our farms that we worked with in the past, those that have, you know, quite a high incidence of yonis. We would suggest that they try to vaccinate their replacers, at least the lambs that are going to be breeders later. And again, there are guidelines as to when best to vaccinate your lambs. But yes, by vaccinating your ewes, that won't protect the lamb.
And would you back, if you thought there was a decent enough issue with yonis, that the farm from start should actually vaccinate the whole flock, including the older ewes? Will you get a return on vaccinating older ewes?
Yeah, so yeah, interesting question, because older ewes don't always are a little bit more resistant to getting infection. And I guess it depends on the extent of the problem and how quickly you want to try and get it under control. So some people will vaccinate their entire flock. Others find that that's just too big an investment, so they might just try to vaccinate the replacer lambs. Yeah.
But if she's already got yonis, but then you vaccinate her at, say, 3 or 4 years old, would that reduce her shedding, or the vaccine do nothing at that point if she's already got it?
Yeah, it's hard to say. So I think the jury's still out on that. I'm not quite sure what the answer is to that. I do know that vaccinate doesn't prevent them from dying from yonis necessarily. So that can be very dispiriting for farmers that we worked with in the past. And they say, well, you know, my ewe died of yonis last year, and I vaccinated it last year. What's going on? And unfortunately, if they're infected before they're vaccinated, that's a problem. But also, as I said before, the vaccine is not a sort of gold bullet. It doesn't stop sheep from getting infected. But it does really reduce it. So that's a good thing. But it won't necessarily stop them from getting clinical disease, especially if they've been infected before they're vaccinated.
Have you looked at other co-infections in addition to worms, i.e. fluke or other iceberg diseases?
Yeah, well, very interesting question. Yeah, so there's been some studies about co-infection with fluke and with MAP, and also with bovine tuberculosis as well. The co-infection between bovine TB and fluke and the effect it has on the diagnostic tests. We would be very interested to try and get a project going in looking at other co-infections. At the moment, we're not really in a position to be able to do that, but we are aware that there are other groups working on co-infections of other pathogens, such as liver fluke, particularly. But also the interesting question of when in cattle, particularly when they're co-infected bovine TB, cattle are infected with maybe with MAP as well. So lots of really interesting questions out there to try and answer.
Certainly is. Yep. I suppose maybe to combine a few questions, also one of my own, about incoming stock. So should we be testing incoming stock? In myself, I was thinking about the tops coming in. You know, should we be testing them? Should we consider vaccinating bought-in tops? You know, we're generally buying them in as a ram lambs or a shearlings. Should we be just buying a little bit of vaccination just for them, even as a bit of an insurance? And a final question on the ram side: does it pass through the semen, the MAP?
Yeah. Okay. So last question first is yes. That's quite a quick one. It's not the main transmission route. As I said, fecal oral is probably the main transmission route. But yes, it will pass through semen as well. If you're bringing frozen semen in, good to make sure that comes from an accredited flock. It's interesting because people will tell me, we have a closed flock, but then they'll be bringing rams in, which is, that's what most farmers will be doing to try and keep their gene pool nicely mixed. And it's important to do that. But it's interesting that they're not always tested. And I would think that it'd be good to at least know where the rams come from, whether they've come from an accredited flock or from a source that has been free of Ionis for a number of years or is vaccinated. And always, if you think it's worth it, then definitely to try and keep Ionis at bay, definitely think about testing these rams as well. So we've come across this before in the past where people are saying, well, we haven't had Ionis before, but now we suddenly realize it's in one of the rams that we brought in. And so that is kind of like a biosecurity gap, I suppose, bringing the rams in. So well worth checking the source and testing before you let them loose with the flock.
And would you use fecal or bloods for a few rams?
So I would probably, again, it's worth speaking to your local vet on this. They'll have an idea as well, but potentially sequentially test the fecal, the fecal test, the qPCR. No harm in trying to do an antibody test. At least it would tell you whether they've had an exposure to the infection in the past as well. The ELISA, the blood test is obviously a lot cheaper, but having a ram on your farm, you really want some really good offspring, and the last thing you need is the Ionis problem. So I would probably if I could afford it and have the time to do it, then I would probably do both tests. Okay. But particularly the qPCR, because it is so sensitive and so specific, but not just a one-shot test, but perhaps a couple, 2 or 3 tests before you let them loose.
How far apart then?
Good question again. So again, that's something that you have to ask your vet. I don't have a magic number how far apart those tests should be given, but we do know that sometimes stress can really bring on Ionis disease and bring on an increase in transmission. So if rams are being transported in, then it's a good opportunity then when they're a bit stressed just to do a test right away. and so I guess that would be a prime time. But then it can be the next week or the next month. I'm not. There doesn't seem a magic number there.
Yeah, great. There's still questions coming in, which is
Great. Oh, wonderful.
We're happy to do a few more minutes. Someone said I'll tie the two questions in. I thought you had to vaccinate sheep as lambs before 6 months of age, and also are sheep susceptible at any age?
So sheep are susceptible at any age, but it has to be said that older ewes are a bit more resistant to infection. There's no doubt about it. So you get older ewes, 4 or 5 years old, much more resistant to being infected than the young lambs. Yeah.
And in terms of vaccination, age, or how young could you go with the vaccination, or when would be appropriate?
Yeah, again, worth asking your vet about that. And looking at the Good Air guidance on that. So it does vary a bit from country to country, but I think something like 6 weeks or something like that seems about right. But do check with your vet, because I'm not entirely sure.
Perfect. And I suppose, just given the cost as well, you'd probably want to know that ewe lamb's probably getting kept on. To put an expensive vaccine in a finishing lamb might become kind of a costly game.
Yeah, I mean that's a big question to ask. I mean, do if you're going to if they're going to be finishers, do you want to vaccinate? And that is a tricky one, because obviously while they're growing up, they could still be producing or shedding bacteria into the soil and around the pasture and spreading it to your flocks while they're still on farm. And the other thing, of course, is the vaccine does cause a bit of a lesion, which can be lifelong, and so sometimes farmers are a bit worried about the fact that you get this big lesion on the neck where the injection site, and I say they're worried about the value of the lamb or the sheep at that point. I think there was a really interesting study in New Zealand about that, and they seem to think that the carcass value didn't go down, but that's in New Zealand. So I don't know what the situation is in the UK.
Perfect. Someone said, are you suggesting that higher levels of stomach worms might adversely affect the Good Year efficacy? If so, would you suggest worming your replacement ewe lambs before administering?
Well, this is what we want to find out. This is exactly what we're trying to find out. So what I can say is we've done some association studies, and we've had some where we've just been doing fecal leg counts at the moment, but we're now in a position where we're able to find out what species particular worms we're dealing with. But we do know that with the clinically affected sheep, you get a much higher egg load, fecal egg count. And you kind of think, oh well, yeah, I suppose that makes sense. The sheep is suffering from one illness. It's going to be more susceptible to other things. But we think there's a lot more to it than that. And one thing is that we know that MAP can be carried actually on the nematode or on the roundworm, and of course the roundworm buries itself into the side of the small intestine. So that's a really good way for MAP to get infected into the gut. But also from an immune system point of view, there's a kind of two kind of directions mostly that the immune system takes, and that's a Th1 or a Th2. And don't worry about the sort of detail about that, but worms cause a Th2 kind of direction of the immune system. And when you get sheep moving from the sort of subclinical, looking fine, healthy, to this clinical phase, the immune system goes in the same direction, goes into that Th2 direction of the immune system. So if you've got worms that are there already that are pushing the immune system in that direction, then we think that that's a possibility that's also driving those sheep that got high worms in their small intestine to also start getting clinical disease. But that is our theory. That's our hypothesis. We're working on trying to find out whether that's we can back that up or not.
Brilliant. We'll just finish on one last question then. Could you maybe just take us through what it would entail to be in a health scheme for the sheep, and also are accredited flocks completely free of it, or are there different risk levels the same as cattle? So we'll finish on that. But thank you everyone for so many questions.
Yeah, no, the question's been fantastic.
Yeah.
So the premium health scheme for sheep and goats, please look that up on the website, because that is really clear about how what are the expectations, and anybody can join it. There is a different stages as well, and expectations on that, and I don't really want to go through the details of it because I'll probably get myself confused with the cattle one, because I often do. Yeah, so I think for the cattle one, it's something like 3 to 5 year clear, or a 3 years of continuously clear tests is the sort of level 1, and then they're accredited. Accredited flocks or herds don't necessarily mean that they are actually Yonis free, but it does give people and buyers and people who are purchasing and selling real confidence that, you know, they have got the disease as under control as they possibly can, and that these animals are as negative as we can manage to do with the tests that we have at the moment. So I think the accreditation schemes are an absolutely excellent idea, and I think the if you can get yourself involved with one of those, then it's really well worthwhile. And also, I mean, Daniel, you mentioned this new sort of program coming out with a sort of £250 worth of to try and incentivize people to do some cull testing, and I think that's a really good way to start as well. So yeah, that's well worth checking up on.
Perfect. No, it's absolutely brilliant. Thank you everyone for all the great questions, and we'll wrap up now, but thank you very much, Craig, for joining us tonight. It's been hugely informative. Certainly enjoyed it.
Thank you. And just to say that, you know, we are at Moredun. We're more than happy for people to get in touch with me at Moredun. Just look up on the website. We have something like 12,000 Moredun members as well throughout the UK. And they all have access to individual scientists should they wish to know what's going on. And we do get quite a few farmers as well as vets phoning us up. So bear that in mind, that, yeah, if you're a Moredun member, you can get in touch with me. But otherwise, do be in touch, and we'll see what we can do. But thank you, Daniel, very much for bringing me on tonight. That's been lovely.
Okay, perfect. Thank you, everyone, and good night.
Good night.